Assalamualaikum warahmatullahi wabarakatuh. Hello, let me introduce ourselves. My name is Valia Rahma from group of 12, and there are my friends in here, namely Oki Rizky Pratama and Sabrina Putri Khairani. Here, I will explain why our group chose to become a pro team on this topic, and our topic is animal genome modifications. Okay, let's get started, and here we go.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Oki Rizky Pratama. Now we will discuss about Animal Genome Project. For the first, we will discuss about what is the Animal Genome Project. Okay, animals models are indispensable for understanding disease pathogenesis and developing novel therapeutic agents and treatments. By improving the availability and quality of optimized animal models, the translation of preclinical studies in model organisms to clinical trials in humans might become more of reality. With the identification of mutation responsible for human disease, target modification of these genes in large animals provides useful disease models for pathology studies, drug discovery and development, and regenerative medicine. With the new livestock technologies beneficial, unless there would be otherwise be lost in conventional breeding could be converted using the novel genome editing tools, and also result it reduced cost and shorten time frame for generating the desired mutant animals. In this presentation, we briefly provide an overview of the development of nuclease-mediated gene editing tools and discuss the representative studies that have been achieved using gene editing tools for modeling human disease, regenerative medicine, and agricultural application. And next, we will discuss about animal genome modification. Genetically engineered animal models that reproduce human disease are very important for the pathological study of various conditions. The development of the cluster regularly interspace palindromic repeats or CRI-SPR system has enabled a faster and cheaper prediction of animal models compared with traditional gene targeting methods using embryonic stem cells. Generation of animal models of human disease using genome editing methods. The use of fertilized one-cell stage embryos is the most common method of producing genome-engineered animal models. The methods that are used for producing animal models using fertilized embryos with the CRI-SPR system include microinjection, electroporation, and genome editing via oviductal nucleic acid delivery or CONADE. The most common animal model of disease generated using genome editing is the rodent, including mice and rats. We can see the picture. Microinjection is a method of injection the CIS9/gRNA complex directly into the cytoplasm of pronucleus of fertilized one-cell embryos, whereas electroporation enables gene editing by including electric stimulation in the presence of the CIS9/gRNA complex to fertilize one-cell embryos. GONAD is a new method to introduce the CIS9-gRNA complex into embryos without sacrificing animals. The CIS9-gRNA complex for genome editing is injected directly into the oviduct of pregnant mouse. followed by in situ electroporation. Here we describe studies of animals, models of disease using various CRISPR-based genome editing methods.
How the Animal Genome Project works. The procedure of conducting a genomic test is as follows: genome testing, informed consent, sample collection, sorting, analysis, interpretation of variants, case review, and interpreting results. Methods used in genetic engineering techniques, vectors, cloning, PCR, or polymerase chain reaction, selection, screening, and recombinant analysis. Next, the steps of genetic recombination include first, identification of the expected gain, introduction of DNA code to the expected gain, regulation of the expression of the engineered gain, and monitoring of gene transmission to offspring. Next, application of Animal Genome Project. The first, glowfish. Glowfish is one example of a genetically engineered transgenic animal. This fish was developed from the United States that engineered DNA from zebrafish, the Neurario, with a gene encoding green fluorescent protein from GFP, or green fluorescence protein. However, phenotypically, the resulting color is not only green but yellow to red. Mosquitoes, a British company Oxitec has created genetically modified male mosquitoes that carry a self-limiting gene. When they are released into the wild and mate with females, their offspring don't reach adulthood, so crucially don't contribute to the spread of the Zika virus. Other researchers are looking at using genetic modification to curb the spread of malaria. Next, and rabbit, bispecific T-cell antibody producer. One of the deadly human disease is cancer. This disease can be overcome by increasing T-cell antibodies. Now, using genetic engineering, rabbits can be used as test animals to produce two kinds of specific antibodies, namely CD28 and R28M, molecules that can encode TCR/CD3 that can kill cancer cells. With the discovery of these bispecific antibodies, it's hoped to gain enough knowledge about bispecific antibodies for medical application. Genetically modified organism, or GMO, in Indonesia, or GMO cattle. GMO cows can produce milk that is similar in nutrients, enzymes, and hormones to breast milk. To production of transgenic cattle is highly dependent on the quality of the one-cell embryos to be injected. If embryos are obtained in vivo, the produce begins with superovulation of donor cattle to obtain many embryos, collection of zygotes, one-cell embryos, microinjection of DNA in embryos, culture or embryo until the blastocyst phase, transfer to recipient animals and obtain transgenic cattle. The utilization of transgenic technology allows the acquisition of livestock with superior characteristics such as transgenic livestock in use as a model to determine the process of disease in humans. Transgenic livestock in use as a way to modify the anatomy and physiology of the livestock itself. Transgenic livestock is specifically made to produce human proteins, which can be enzymes or hormones.
Okay, next, I like to mention that there are many points here. Firstly, in our opinion, do the benefits of genetically modified farm animals outweigh the risk? Okay, genetic engineering holds great potential in many fields, including agriculture, medicines, and industry. And genetic modification can increase the yield from farm animals. For example, cows can be engineered to produce more milk for the same size of herd. Okay, next, genetically modified farm animals are being used to produce important medicinal products such as antibodies in large quantities, and these products can be used for the treatment of many different human conditions. The current production system for such products is donated from human blood, which is in limited supply due to lack of donors. Okay, sheep and goats can be modified to produce medicinal products in their milk. This has no negative impact on the animal, but the product can help to treat human disease. Okay, secondly, is genetic modification of farm animals ethical? Genetic engineering is a logical continuation of selective breeding, which has been done by humans for thousands of years. Genetic engineering of animals is strictly controlled by animal cruelty legislations in many countries and is always carefully scrutinized by teams of experts before being approved for wider use. Next, is it morally acceptable to genetically modify farm animals? Animals have been used to help humans for millennia. Many would say that human lives are of higher moral value than animal lives. The percentage of genetically modified farm animals is tiny compared to the number of animals slaughtered for humans to eat. This practice is widely seen as morally acceptable. Okay, next, is there a true regulatory process for genetic modification of farm animals? There has been suggested that, as a rule, genetically engineered animals should be no worse off than the equivalent stock would be if they were not genetically engineered. First, the Animal Welfare Act, a federal law passed in the UK in 1966, requires all entities looking to carry out research with animals to have their program reviewed before they go ahead, have veterinary care programs in place, and staff that are qualified to care for live animals. Second, the UK Home Office Animals or Scientific Procedure Act in 1986 has set out guidelines for research involving animals. Okay, the FDA or Food and Drug Administrations in the USA regulates genetic engineering with animals and their products under the new animal drug provisions of the Federal Food, Drug, and Cosmetic Act. It helps monitor and maintain certain standards, including input from the public when it comes to genetically engineering animals. The new animal drug provisions of the FFDCA focus on whether the new animal drug is safe for the animal and if it is effective. If the drug is for a food-producing animal, it also focuses on whether the resulting food is safe to eat. Okay, next, government regulatory number 39 year of 1995 about research and health development. Government case number 72 and case per to 1999 about implementation of artificial reproductive technology, and many more. Okay, based on explanations, next opinion is genetic modification proposed to save endangered species forming superior individuals. GMOs may have more nutrients, eliminate disease in animals. The last is conclusions. Let me summarize our point. In summary, the rapid progress of genome editing in large animals has resulted in the production of many valuable animals for human disease models, xenotransplantations, and the agricultural economy. Further optimizations of the existing genome editing system and the generations of new tools for precise gene modification will additionally accelerate the development of genetically modified animals, organs, and tissues for agriculture, regenerative medicines, and therapeutic applications.